This recording is Chapter 4, Part 3 of 3, The Tissue Level of Organization. Cartilage is a supportive connective tissue. The matrix of cartilage contains a lot of chondroitin sulfate, which will combine with other proteins to form molecules known as proteoglycans. Proteoglycans have a tendency to attract water molecules. That's why cartilage is made up of 80% water. Proteoglycans are also helpful in and of the fact that they help the cartilage resist stress and compression. The cells in cartilage are called chondrocytes. Chondro meaning cartilage, cyte meaning cell. They reside in special little sites called lacunae, and cartilage is avascular, which means it does not have a good blood supply. Therefore, the chondrocytes have to get nutrients and oxygen by way of diffusion, and they also have to get rid of their waste products and carbon dioxide by way of diffusion. Cartilage lacks nerve fibers and is surrounded by dense irregular connective tissue that forms an outer layer surrounding the cartilage that is known as the perichondrium. And the perichondrium not only protects the cartilage, but it's important for cartilaginous growth. There are three types of cartilage in the human body. We have hyaline cartilage, elastic cartilage, and fibrocartilage, sometimes referred to as fibrous cartilage. Hyaline cartilage is the most abundant type of cartilage in the human body. We have hyaline cartilage between our ribs and our sternum, on the ends of bones in synovial joints, as well as part of the nasal septum is formed by hyaline cartilage. We see hyaline cartilage in parts of the larynx, which is the voice box, making up the rings of the trachea as well as the bronchi. Hyaline cartilage provides stiff but flexible support as well as reduces friction between bony surfaces. When we look at hyaline cartilage under the microscope, one of the things you'll notice is that when we look at the chondrocytes in the lacunae, the surrounding matrix, the area around the lacunae, has a very smooth appearance, which is sometimes referred to as a ground glass appearance. If you ever look at a piece of glass that has been tumbled around in the waves on the beach, it has a very smooth ground glass appearance, and that's how the matrix of hyaline cartilage is often described. Elastic cartilage is found in the auricle of the ear. That's this part of the ear. You can see how flexible it is. It's also found in the auditory tube, the epiglottis, and the cuneiform cartilages of the larynx. Elastic cartilage will provide support. However, it also can allow for distortion without tissue damage and will return to its original shape. When we look at elastic cartilage under the microscope, you'll notice elastic fibers in between the chondrocytes that are located in their lacunae. Fibrocartilage is found in the menisci of the knee, which are pads within the knee joint, between the pubic bones of the pelvis, forming the symphysis pubis, as well as the intervertebral discs between the bones of the spine, the vertebrae. Fibrocartilage has a lot of collagen fibers, thick collagen fibers, that will resist compressive forces and also prevent bone-on-bone contact while limiting movement between the bones relative to one another. When we look at fibrocartilage under the microscope, you'll notice the wavy, thick collagen fibers between the chondrocytes located within their lacunae. By the way, lacunae is plural for lacuna. Vertebrae is plural for vertebra. Cartilage forms in two ways. Interstitial growth is going to occur when chondrocytes undergo mitosis. After chondrocytes undergo mitosis, with that parent cell dividing into two identical daughter cells, as the daughter cells separate, they begin to form their own individual lacunae and will secrete cartilaginous matrix around them. As they grow, they're expanding the cartilage from within. That is interstitial growth. We mentioned the fact that outside cartilage is a layer of dense irregular connective tissue. The dense irregular connective tissue contains fibroblasts, but it also has dividing stem cells within it. Those dividing stem cells in the perichondrium will differentiate to become chondroblasts on the outside of the cartilage. Those chondroblasts will secrete cartilaginous matrix around themselves. And as the matrix increases, more chondroblasts are going to be incorporated in this matrix, and they are going to be replaced by divisions of more stem cells in the perichondrium. So appositional growth is occurring on the outside, making it larger. Interstitial growth is occurring on the inside, increasing the size. Whenever you see a cell that says blast, blasts create that type of tissue or more cells. Fibroblasts make fibers. Fibrocytes maintain the fibers after they're made. Chondroblasts secrete cartilage matrix. Chondrocytes maintain the matrix after it's been secreted. Osseous connective tissue is probably something you're more familiar with when you hear the word bone. Osseous connective tissue comes in two types. We have compact bone and spongy bone. Compact bone is going to be the type of bone that makes up the shaft of a long bone or the outside of a flat bone. Spongy bone is found in the ends of long bones and the middle of flat bones, and is also the majority of what's in the vertebrae. The matrix of osseous connective tissue is going to contain a lot of calcium salts in the form of a molecule called hydroxyapatite. Calcium salts make up 2/3 of the matrix of osseous connective tissue, which contribute to the fact that it is hard. However, osseous connective tissue also contains collagen fibers. Remember, collagen is the most abundant protein in the human body. The collagen fibers allow bone to be somewhat flexible, and they lend to the strength of bone. So calcium salts make it hard, collagen fibers make it strong. You'll notice in this picture on the right-hand side that bone has a periosteum, which is 2 cell layers thick. The outer fibrous layer and the inner cellular layer are what make up the periosteum surrounding bone. Bone, unlike cartilage, is highly vascular. We already said there's a periosteum on the outer surface There is also an inner layer on the inside of osseous connective tissue called the endosteum. That word is actually written on this slide on the upper left-hand side. It's just very small. On this picture down here on the left, we can see compact bone arranged in these units called osteons. And there is a little bit of spongy bone, which is also referred to sometimes as cancellous bone, on the inside of the shaft of the long bone. We'll look at that a little bit more closely when we actually get to the skeletal system. So osteons are the structural units of compact bone. Osseous connective tissue contains osteocytes that also reside in special spaces referred to as lacunae. Because osteocytes reside in a matrix that is very, very hard, they have special little canals referred to as canaliculi that allow different substances to diffuse between the blood supply and other osteocytes. That allows for exchange of materials and substances. This takes us to fluid connective tissues, which include blood and lymphatic fluid. Blood is found in blood vessels as well as in the heart, and it's responsible for carrying nutrients and waste to and from the cells of our body. The cells found in blood are scattered in a fluid portion of blood referred to as plasma. There are different types of cells that are found in blood, including erythrocytes, which are red blood cells. They look like the little red donuts that you see in the slide above. We also see leukocytes, which are white blood cells, and there are different types of white blood cells, as well as platelets and plasma proteins. Fibers are only present in blood when a tissue injury occurs that starts a cascade of clotting reactions that will cause different cells to produce fibers to stop the bleeding and form a blood clot. Lymphatic fluid is formed from interstitial fluid that enters lymphatic vessels, and this helps to carry excessive fluids and proteins back to the blood, because the lymphatic system actually ends up dumping into the cardiovascular system. The lymphatic fluid is a part of our immune system, and in this particular slide we see a picture of a lymph node. Let's see if we can name the connective tissues on these slides. We actually have dense regular connective tissue in the slide at the upper left, right here. We have dense irregular connective tissue in the middle slide, and elastic connective tissue on the upper right. Did you guess that this is the reticular connective tissue that looks like a bunch of grapes or cherry blossoms? Here we have fibrocartilage in the center. This slide we haven't yet looked at. This is adipose connective tissue. It contains special cells called adipocytes. The nuclei tend to get pushed toward the periphery of the adipocytes because they're filled with fat. Here we have osseous connective tissue that you can recognize by the osteons, which look very similar to tree trunks. Here we have areolar connective tissue in the bottom middle, appearing like spaghetti and meatballs. And this is actually elastic cartilage. It's tough to see. There are tiny little elastic fibers on that picture. As we mentioned previously, connective tissue is important because it helps to form the body's framework. And there are actually layers of connective tissue within the ventral body cavity alone. You can see all of these different layers of fascia that provide strength and stability. And also, we know that internally there are different structures of connective tissue, such as internal ligaments, that help to maintain the position of internal organs. Connective tissue can also provide a route for blood and lymphatic vessels as well as for nerves. Looking at this longitudinal section of the ventral body cavity, you can see there's many layers of fascia or connective tissue in between the layers of muscle and skin and fat. You do not have to know those names of the fascia in between everything. It just is a good illustration of how much connective tissue and how many layers there are within the body that help hold things together. Here's another picture, and sometimes those layers are referred to as superficial fascia, deep fascia, and subserous fascia. Again, I'm not concerned with you knowing those names and where they're located, but obviously you can see these different layers that are going to help connect things. And the superficial fascia is actually made up of areolar and adipose connective tissue, whereas the deep fascia is made up of dense connective tissue. It's actually dense irregular connective tissue, whereas the subserous fascia is going to be between serous membranes and deep fascia and contains areolar connective tissue. That's too much minutia and much more information than I need you to know for your exams. Just really want you to know that there are so many different layers of connective tissue involved in holding things together. Muscle tissue is one of the 4 major tissue types in the human body. Remember, we have epithelial tissue, connective tissue, muscle tissue, and nervous tissue. We have 3 types of muscle tissue, including skeletal muscle tissue, cardiac muscle tissue, and smooth muscle tissue. When we look at skeletal muscle tissue first, we see that the cells of skeletal muscle are long and cylindrical. They're also striated, which means they've got these lines that run perpendicular to the length of the skeletal muscle fiber. Skeletal muscle forms by myoblasts fusing together to form one long cylindrical muscle fiber. That's why skeletal muscle is multinucleated. The nuclei of those myoblasts, when they fuse together, are going to be incorporated in that one long cylindrical muscle fiber. Skeletal muscle tissue combines with connective tissue and nervous tissue and is important because not only does it move our skeleton, it also helps to stabilize our joints so that we can have more effective motion. Skeletal muscle tissue also helps us produce body heat and protects our internal organs. It's also important because it guards the entrances and exits to the digestive, respiratory, and urinary tracts. Skeletal muscle is striated, it is multinucleated, and it is voluntary. That means we have to generate a thought in order for skeletal muscle to go into action. If I want to walk backward, I have to generate the thought to walk backward. Same thing if I want to walk forward, if I want to raise my hand. Cardiac muscle tissue is made up of cells that are short, that tend to branch, are also striated with lines that run perpendicular to the length of the cell. However, they tend to have one, maybe two nuclei. Usually you just see one nucleus with cardiac muscle. Cardiac muscle tissue also has very important cell junctions. These cell junctions are referred to as intercalated discs. You'll remember from our discussing cell junctions earlier that intercalated discs include desmosomes, which are those spot welds or Velcro that hold things together tightly, and gap junctions, which allow ions to flow freely from one cell to the next. The gap junctions allowing ions to flow help the heart beat in what is known as a functional syncytium. Basically, it functions in sync. There are 4 chambers in the heart. The 2 upper chambers are the atria. The 2 lower ventricles, the 2 lower chambers are the ventricles. The heart beats atria, ventricles, pause, atria, ventricles, pause, atria, ventricles, pause. That's the functional syncytium that is allowed by the intercalated discs as well as the electrical conduction system of the heart. When we look at the slide, you'll notice these dark red lines are where the intercalated discs are located. Obviously, cardiac muscle tissue is only going to be found in the heart, and it helps to circulate blood throughout the body, and that helps to maintain blood pressure, which is also known as hydrostatic pressure. This takes us to smooth muscle tissue. Smooth muscle tissue consists of short cells that are spindle-shaped and tapered on the ends. They come to a little V on either end. And these cells have nuclei that tend to be elongated, oval-shaped. Smooth muscle tissue is going to be found in the walls of hollow organs, such as blood vessels, the trachea, bronchi, the intestines, the stomach, the urinary bladder. So we see it in the walls of hollow organs throughout our body, except the heart, because that has special muscle tissue. And the functions of smooth muscle are going to be to move food, urine, reproductive secretions, as well as helping to control the diameter of respiratory passageways, as well as the diameter of our blood vessels. When we look at smooth muscle tissue under the microscope, you can see those oval cigar-shaped nuclei in smooth muscle. Now, skeletal muscle, we said, was striated, multinucleated, and voluntary. Cardiac muscle tissue and smooth muscle tissue are involuntary. You do not have to think beat, 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 or have your intestines go short and squeeze, short and squeeze, short and squeeze. So skeletal muscle of the 3 is the only one that is voluntary. The other 2 are involuntary. Skeletal muscle and cardiac muscle are striated, where smooth muscle is not. And skeletal muscle of the 3 is technically the only one that is multinucleated. Can you tell which muscle type we're looking at on this slide? The intercalated discs really stand out on the slide on the upper left. Therefore, we know this is cardiac muscle. These long cylinders that appear like worms because the striations are visible are going to be indicative of skeletal muscle, and the elongated oval nuclei down below in the bottom slide are very common with smooth muscle. This takes us to nervous tissue. Nervous tissue includes neurons and neuroglial cells. Neurons are the cells that are most commonly associated with nerve impulse conduction. Neuroglial cells are the supporting cells that support neuron function. When we look at neurons, we see that they have a cell body which contains the nucleus and other major organelles. The cell body of the neuron is often referred to as soma. We see that extending off of the soma of the neuron are extensions that are referred to as dendrites. The dendrites are responsible for receiving incoming information, and the soma of the cell body will be responsible for receiving incoming information. You'll notice that there is an extension going away from the soma, which is referred to as the axon. The axon conducts information away from the soma to other cells. We always used to say axons away. If you think of a neuron, you can think of a hand with the bulk of your hand being the soma or the cell body, your fingers being the dendrites, and if you're catching information, you use that whole hand, and then the axon takes information away. At the end of the axon, you'll notice that there are little branches, and we'll discuss those, and there are little bulbs at the ends also. We'll discuss those in greater detail when we get into the nervous system. So the axons are the main cells in nervous tissue responsible for receiving and sending information in the form of an electrical impulse. The term neuroglial literally means nerve glue. These are the supporting cells that help to maintain the physical structure of nervous tissue. They're also important in repairing tissue and that nervous tissue framework after an injury occurs, and they can perform phagocytosis, eating up debris, and getting rid of bacteria and pathogens. They're responsible for providing nutrients to neurons as well as regulating the composition of ions in the interstitial fluid, the fluid between the cells, so that neurons don't fire inappropriately. Our nervous system is broken down into two different areas. The central nervous system includes the brain and the spinal cord, whereas the peripheral nervous system includes the nerves that go to the rest of the body to and from the brain and spinal cord. The peripheral nervous system includes two types of neuroglial cells. We've got satellite cells, which are going to wrap around the cell bodies in special collections of cell bodies in the peripheral nervous system known as ganglia, and they're responsible for regulating the concentration of oxygen, carbon dioxide, as well as neurotransmitters around the neuron cell bodies within the ganglia. Also involved in the peripheral nervous system are cells known as Schwann cells. Schwann cells wrap themselves around the axons in the peripheral nervous system, creating a special covering called a myelin sheath that allows for faster nerve impulse conduction. It's kind of like the wiring on an electrical wire. The Schwann cells can also help repair injured axons if they've been damaged. There's a limited amount of repair that they can do, but they can help in that process. In the central system, we've got more neuroglial cells, including oligodendrocytes, which will produce myelin around axons in the central nervous system and help provide structural framework. We also see astrocytes, which are star-shaped cells, astro meaning star. These astrocytes are really important because they help to maintain the blood-brain barrier. They provide structural support. They help to regulate the concentration of ions, nutrients, and dissolved gas concentrations in the extracellular fluid around neurons. They also help to absorb and recycle neurotransmitters and form scar tissue after injury has occurred. Microglial cells are tiny little cells that remove cellular debris, waste, and other pathogens by way of phagocytosis. And ependymal cells are special cells that line spaces inside the brain known as ventricles that produce cerebrospinal fluid. And the ependymal cells play a role in producing cerebrospinal fluid and also in circulating and monitoring the concentration of ions within that fluid. Let's see if we can name these tissue types. Here we have skeletal muscle on the upper right, cardiac muscle on the lower left. You can see the intercalated discs, smooth muscle on the upper left picture, and nervous tissue on the lower left. By the way, you can see the neuron cell bodies, the soma with their dendrites in this picture, and sometimes you can distinguish which one of those extensions is the axon. We also see these dark nuclei of different neuroglial cells in this slide. When tissue injury occurs, that exposes tissues to pathogens or toxins, and mast cells are going to be stimulated to release inflammatory chemicals such as histamine and heparin. Remember that we said histamine is going to cause vasodilation and increased capillary permeability, where heparin is going to prevent blood from clotting. That means inflammation is going to cause an increase in blood flow, and as the blood vessel permeability increases, more fluid can leak out of those capillaries into the surrounding area, causing swelling and pain. So with injury, we see that there is an increase in tissue concentrations of pathogens, toxins, waste products, as well as chemicals from the cells that have been damaged or injured. That activates mast cells to not only release histamine and heparin, we're also going to see the release of chemicals called prostaglandins. You'll remember that prostaglandins are a type of lipid molecule that will send pain signals. There are different prostaglandins through the body, and some actually help protect the lining of the stomach. In this case, they're going to be for pain signals. With the histamine, we have increased blood flow through vasodilation and increased capillary permeability. The heparin is going to prevent blood from clotting, and prostaglandins are going to send signals that there's pain. This will cause an increase in local temperature or heat, increased flow of oxygen and nutrients to the damaged area, as well as an increase in the number of special cells known as phagocytes. they're going to eat up the parts of the tissue that have been damaged. They can also remove the toxin or waste products that have been created by the injury. And the inflammation will eventually lead to regeneration, where we have decreased concentration of pathogens, toxins, waste products, and chemicals from the injured cells, and a restoration of normal tissue conditions. We're going to look at tissue injury first by looking at what happens in a non-extensive skin wound. If we have a simple cut into the dermis, the severed blood vessels are going to start to bleed, releasing inflammatory chemicals, which we already know are histamine, heparin, and prostaglandins. And those local blood vessels will become more permeable, allowing fluid to go through, white blood cells to go into the area, as well as clotting proteins are going to start forming a cascade of chemical reactions that will form protein fibers and other plasma proteins that are going to seep into this injured area. So as clotting occurs with the formation of these protein fibers being produced, that's going to form a scab on the outer surface of this wound. And what happens next is the fibroblasts inside the dense irregular connective tissue are going to produce a special tissue known as granulation tissue. And this granulation tissue is going to release chemicals. The cells within the granulation tissue release chemicals that are going to send signals for blood vessels to come back into the area, growing new capillaries. So they're going to restore blood vessels in the area, and that will stimulate fibroblasts to produce more collagen fibers that help to knit the edges together. And as the fibroblasts help to knit that damaged tissue area together, the macrophages that have come in are going to eat up the dead, dying cells and other debris that was there from the injury, and the epithelial tissue on the surface is going to be stimulated to multiply by way of mitosis to fill in to repair the damage that occurred at that level. So the surface epithelial cells are going to migrate over the granulation tissue. That process leads us to the next portion of regeneration, where the area that has a lot of these fibers, which includes a portion of the epithelium as well as the dermis itself, the fibrosed area is going to mature and start to contract into itself as the epithelium thickens. And we often see fully regenerated epithelial tissue with underlying areas of scar tissue as a result, depending on the extent of the damage. We may or may not have a scar. Scarring is not always an inevitable part of the process. So this is a self-limiting response, where once it's done, it's completed. If a person ever rips off a scab and notices there's a little white fiber trying to hold on to it, that's actually the fibers that were produced by the fibroblasts. And that's why it's important not to tear off a scab, but to let it fall off, as will happen as this tissue contracts and the epithelium is regenerated. The process we just discussed is known as healing by first intention. Healing by first intention occurs when the body can take the two edges that have been separated by damage and they're knit together. That is healing by first intention. Sometimes people have wounds that occur from within where tissue dies underneath, such as an ulcer, or sometimes a large wound may occur. And a large wound, such as an ulcer or a big wound like occurred on my thumb, is going to require something called healing by second intention, where a much larger scab occurs and the wound edges remain separated. So the inflammation that occurs on a larger wound or an ulcer is going to be much more intense because there's a lot more dead tissue, and necrotic debris is dead tissue. There's also fluid weeping, which we refer to as exudate, and fibrin that is going to be involved in the process of healing by second intention. So we see swelling and exudate and fibrin working from the bottom to help repair the tissue underneath. So we've got healing by second intention, means that it's going to come and work its way basically from the bottom up. So we see a lot more granulation tissue. That means a larger defect in the final product. We've got slower healing, more scar tissue, and the wound will eventually contract underneath the epithelial tissue. It's just going to take a much longer time to happen. So this is healing by second intention, which mine isn't finished. Tissue regeneration or tissue repair is dependent upon different factors. Epithelial tissue, while avascular, lies close to tissues that have a good blood supply and gets its oxygen and nutrients and gets rid of carbon dioxide and waste by way of diffusion. Epithelial tissue has stem cells which tend to regenerate often. So epithelial tissue has a really good ability to regenerate itself very well. Osseous connective tissue has a great blood supply, therefore it can regenerate well, as does areolar connective tissue and dense irregular connective tissue. They've got a great blood supply. They can regenerate very well. Dense regular connective tissue does not have a good blood supply. It's a little bit worse than smooth muscle tissue. So between the two, smooth muscle tissue will regenerate a little bit better than dense regular connective tissue will. They both have a moderate capacity. Skeletal muscle tissue and cartilage have a relatively weak ability to regenerate. Skeletal muscle would be a little bit better than cartilage. Cardiac muscle tissue and nervous tissue repair poorly, if at all, simply because those cells do not undergo mitosis. And when we have cells that can't regenerate, they are replaced with by scar tissue, which is fibrotic tissue. So we have fibroblasts replacing where the cells would have been with fibers, and that is the process of fibrosis or creating scar tissue. When tissue development occurs, after birth occurs, the cells of most tissues in the body are going to continue to divide until a person forms into an adult. At that point in time, cellular division is really going to slow down. In adults, only epithelial and blood tissues are highly mitotic. Epithelial tissues have to keep regenerating because we constantly wear them down or they wear off. blood cells only live for 120 days, so we have to continuously produce more blood cells. Remember, once red blood cells are mature, they've extruded or kicked out their nucleus. Mature red blood cells can't divide, but red blood cells are regenerated in bone marrow, and when they become mature, that's when they kick out their nucleus. There are other tissues that will regenerate throughout our life, such as liver cells, intestinal cells from stem cells can divide as needed. So there are some stem cells in different areas of our body. We actually have some stem cells in our nasal epithelium. And so they'll divide as needed. As far as aging goes, when people age, the epithelium becomes thinner and is more easily damaged, and the ability to repair tissue damage declines. It doesn't occur as quickly, it's not as effective, and also circulation throughout people's bodies as they get older decreases. If there's decreased circulation, that means that oxygen and nutrients that are needed for repair are not being carried to those damaged tissues. When people age, they tend to stick to a diet that is easier for them to chew, and those softer foods may lack in proteins and vitamins that they need for strong, healthy tissues. Also, cancer rates tend to increase as people get older, so cells are undergoing mitosis that is unchecked and unstopped, creating tumors.